Hello friends, this video is about enzymes. The presentation is being prepared based on Wikipedia, OpenAI and Bioprocess Engineering by Schuler Kargi book. So now, enzymes, there are more than 2000 enzymes in the world as per the second edition of the book. Enzymes are usually proteins of high molecular weight, 15,000 Dalton, several million Dalton, that convert a substrate to a product at a faster rate or catalyzes the reaction without getting converted in the process. There are databases for references. Enzyme commission number or name will be needed for studies. Enzymes are specific, versatile and very effective biological catalysts, resulting in much higher reaction rates as compared to chemically catalyzed reactions under ambient conditions. Some RNA molecules are shown to catalyze reactions and they are called ribozymes. The sites where substrate bind and a reaction takes place is known as active site. The enzyme commission number, EC number, is a numerical classification scheme for enzymes based on the chemical reactions they catalyze. As a system of enzyme nomenclature, every enzyme commission number is associated with a recommended name for the corresponding enzyme catalyzed reaction. An enzyme containing a non-protein group is called a holoenzyme. The protein part of such enzyme is called apoenzyme. Holoenzyme is equal to apoenzyme plus cofactor. International classification of enzymes. So as per Wikipedia, this screenshot has been taken. The International Union of Biochemistry and Molecular Biology have developed a nomenclature for enzymes. Each enzyme is described by EC, followed by a sequence of 4 numbers which represent the hierarchy of enzymatic activity, from very general to very specific. That is, the first number broadly classifies the enzyme based on its mechanism, while the other digits add more and more specificity. The top level classification is EC1 represents oxidoreductases, EC2 represents transferases, EC3 represents hydrolases, EC4 represents liases, EC5 represents isomerases, EC6 represents ligases, and EC7 represents translocases. So OTHLIL T. So the oxidoreductases catalyze oxidation reduction reactions. Transferases transfer a functional group. Hydrolases catalyze the hydrolysis of various bonds. Liases cleave various bonds by means other than hydrolysis and oxidation. Isomerases catalyze isomerization changes within a single molecule. Ligases join two molecules with covalent bonds. Translocases catalyzes the movement of ions or molecules across membranes or their separation within membranes. How to read enzyme classification number? Class first digit. The first digit describes a general type of reaction catalyzed by the enzyme. Oxidoreductases catalyzes oxidation reduction reactions. 2. Transferases transfer functional groups between molecules. 3. Hydrolases catalyzes hydrolysis reactions. 4. Liases catalyzes the removal of groups from substrates without hydrolysis. Isomerases catalyzes isomerization changes within a molecule. Ligases 6. Join two molecules with covalent bonds, often using energy from ATP. Subclass second digit. This digit provides more specific information about the enzyme substrate and the type of reaction it catalyzes within the enzyme class. Subclass third digit. This digit further specifies the enzyme substrate or the type of reaction within the subclass. Serial number fourth digit distinguishes between different enzymes that fall into the same subclass. For example, let's take EC 2.1.1.X. Class 2 represents transferases. Subclass 1 represents acyl transferases. Subclass 1 represents methyl transferases. So ketone. What is a ketone? Just for recalling, ketone is in a carbonyl ketone. The carbonyl group is directly attached to the two carbon atoms, RC double bond O R dash, where R and R dash are alkyl or aryl groups. Acyl groups. An acyl group, on the other hand, is derived from an organic acid by removing the hydroxyl group from the carboxyl group. The general structure of an acyl group is R dash C double bond O, where R dash is an alkyl or aryl group. So this is the difference between the acyl group and the ketone group. So next enzyme entry 2.1.1.39, that is inositol 3-methyl transferase. That is an example. And EC1, if you go for oxidoreductases, what are the different types of oxidoreductases are there? So these are the different types of oxidoreductases which are there. So 1.1, 1.2, 1.3, each one represents one sort of oxidoreductase. And EC 1.1 category, like 1.1.1 means NAD plus or NADP plus as acceptor. EC 1.1.2 means cytochrome as acceptor. EC 1.1.3 means oxygen as acceptor. So 1.1.4 means disulfide as acceptor. EC 1.1.5 means quinone as similar compound as acceptor. 1.1.99 means with other acceptors. So transferases, EC2. In biochemistry, a transferase is any one of a class of enzymes that catalyze the transfer of a specific functional groups. Example, methyl or glycosyl group from one molecule called the donor to another, the acceptor. They are involved in hundreds of different biochemical pathways throughout biology and are integral to some of life's more important processes. Mechanistically, an enzyme that catalyzes the following reaction would be a transferase. X R plus Y is equal to X plus Y R. In the above reaction, where the dash represents a bond, not a minus sign, X would be donor and Y would be the acceptor. R denotes the functional group transferred as a result of transferase activity. The donor is often called coenzyme. So EC2.X. So here EC number 2.1 is methyl transferase. So methyl and formyl transferase. So single groups transferred are single carbon groups. EC2.2 is transketolase and transaldolase, aldehyde or ketone groups. Acyl transferase. Acyl groups are groups that become alkyl groups during transfer. Glycosyl transferase, hexosyl transferase and pentosyl transferase. Glycosyl groups as well as hexoses and pentoses are transferred. Similarly, other sort of transferases work. EC3.X, hydrolases. In biochemistry, hydrolases constitute a class of enzymes that commonly function as biochemical catalysts that use water to break a chemical bond. AB plus H2O gives AOH plus BH. So this typically results in dividing larger molecules into smaller molecules. Some common examples of hydrolyzed enzymes are esterases, including lipases, phosphatases, glycosidases, peptidases and nucleodases. Esterases cleave ester bonds in lipids and phosphatases cleave phosphate groups of molecules. An example of crucial esterase is acetylcholine esterase. Hydrolyzed enzymes are important for the body because they have degenerative properties. In lipids, lipases contribute to the breakdown of fats and lipoproteins and other larger molecules into smaller molecules like fatty acids and glycerol. Fatty acids and other small molecules are used for synthesis and as a source of energy. Classification. Classification 3.1 is ester bonds, 3.2 is sugars, 3.3 is ether bonds, 3.4 is peptide bonds, 3.5 is carbon nitrogen bonds. Hydrolyses are classified. Hydrolyses can be further classified into several subclasses. These are the subclasses: esters, sugars, ether bonds, peptide bonds, carbon nitrogen, acid anhydrides, carbon-carbon bond, halide bonds, phosphorus nitrogen bonds, sulfur nitrogen bonds, etc. And next, if we go to lyases, lyase is an enzyme that catalyzes the breaking of various chemical bonds by means other than hydrolysis and oxidation, often forming a new double bond or a new ring structure. The reverse reaction is also possible. For example, enzyme that catalyze this reaction would be lyase, ATP to C AMP, that is adenosine triphosphate to C cyclic adenosine monophosphate plus phosphoanhydride. So systematic names are formed as substrate group lyase. Common names include decarboxylase, dehydratase, aldolase, etc. Lyases are classified as EC4 in the EC number classification of enzymes. Lyases can be further classified into 7 subclasses. Decarboxylases, dehydratases, carbon nitrogen bonds, carbon sulfur bonds, carbon halide bonds, phosphorus oxygen bonds, and ferrochetolase. Isomerases EC5.X. In biochemistry, isomerases are a general class of enzymes that convert a molecule from one isomer to another. Isomerases facilitate intramolecular rearrangement in which bonds are broken and formed. Isomerases catalyze changes within one molecule. They convert one isomer to another, meaning that the end product has the same molecular formula but a different physical structure. Isomers themselves exist in many varieties but can generally be classified as structural isomers or stereoisomers. Structural isomers have a different ordering of bonds and or a different bond connectivity from one another, as in the name of hexane, and its four other isomeric forms: 2-methylpentane, 3-methylpentane, 2,2-dimethylbutane, 2,3-dimethylbutane. Stereoisomers have the same ordering of individual bonds and the same connectivity, but the three-dimensional arrangement of bonded atoms differ. For example, 2-butene exists in two isomeric forms, cis-2-butene and trans-2-butene. The subcategories of isomerases containing racemases, epimerases, cis-trans isomers are examples of enzymes catalyzing the interconversion of stereoisomers. Intramolecular liases, oxidation reductases, and transferases catalyze the interconversion of structural isomers. So next is 6 is ligases. In biochemistry, ligase is an enzyme that catalyze the joining ligation of two molecules by forming a new chemical bond. This is typically via hydrolysis of a small pendant chemical group on one of the molecules, typically resulting in the formation of new carbon-oxygen, carbon-sulfur, or carbon-nitrogen bonds. For example, DNA ligase can join two complementary fragments of nucleic acid and by forming phosphodiester bonds and repair single-stranded breaks that arise in double-stranded DNA during replication. In general, ligase catalyzes the following dehydration reaction, thus joining molecules A and B. OH plus BH gives AB plus H2O. The naming of ligases is inconsistent, and so these enzymes are commonly known by several different names. Generally, the common names of ligases include the word ligase, such as in DNA ligase, an enzyme commonly used in molecular biology laboratories to join together DNA fragments. However, many common names use the term synthetase or synthase instead, because they were used to synthesize new molecules. There are also some ligases that use the name carboxylase to indicate that the enzyme typically catalyzes a carboxylation reaction. Classification. Ligases are classified as EC6 in the enzyme classification number. Ligases can be further classified into 6 subclasses. EC6.1, carbon-oxygen bonds. 6.2 is carbon-sulfur. Next is carbon-nitrogen, carbon-carbon, phosphodiester, and nitrogen metal bonds. Finally, translocase. Translocase is the enzyme. The reaction most translocase catalyze is AX plus B side 1 equals AX A plus X plus B side 2. So a clear example of an enzyme that follows the scheme is a hydrogen plus transporting 2 sector ATPase. ATP plus H2O plus 4H plus side 1 gives ADP plus phosphate plus 4H plus side 2. So this ATPase carries out the dephosphorylation of ATP into ADP while it transports hydrogen plus ion to the other side of the membrane. So when it comes to the history, the enzyme classification was first approved by India International Union of Biochemistry in 1961. 6 enzyme classes had been recognized based on the type of chemical reaction catalyzed, and several of these involve hydrolysis, as discussed previously. The 7th one was added very recently. So purification of cells and enzymes. So cell purification technique. Cell purification technique involves a tissue dissection. First in tissue dissection, first we have to harvest the tissue, minimize contamination, and then mechanical disruption or enzymatic digestion. Obtain the tissue from containing the cells of interest. This could be from organs, tumors, or other biological samples. Minimize contamination. Work in a sterile environment to minimize contamination. Use sterile instruments and handle tissues with care. Mechanical disruption or enzymatic digestion. Mechanical disruption. Use a homogenizer, grinder, or other mechanical methods to break the tissue into smaller pieces. This is suitable for tissues with less extracellular matrix. Enzymatic digestion. For tissues with a significant extracellular matrix, enzymes like collagenase, trypsin, or DNase may be used to digest the matrix and release individual cells. Filtration or sieving. Filtration. Pass the cell suspension through a filter to remove larger tissue fragments and obtain a single cell suspension. Sieving. Use a mesh or sieve to separate cells from undigested tissue components. This is particularly useful for softer tissues. Cell purification technique continued. Centrifugation, pelleting, washing and resuspend and viability and cell counting. So centrifugation, density gradient centrifugation, layer the cell suspension on a density gradient medium and centrifuge. This separates cells based on their density. Pelleting, so centrifuge the cell suspension to pellet the cells at the bottom of the tube while leaving unwanted debris in the supernet. Washing and resuspension, wash the cells, remove debris, enzymes or other contaminants by washing the cell pellet with a suitable buffer or medium. Resuspend the isolated cells in a medium or a buffer appropriate for their survival and intended downstream applications. Viability and cell counting, use a dye such as tryptophan blue to distinguish between live and dead cells. Cell counting, count the cells using a hemocytometer or an automated cell counter. The method chosen for cell isolation can vary based on factors such as tissue type, cell fragility, and the downstream applications. Cell isolation is a crucial step in various fields, including cell biology, regenerative medicine, and cell culture for research or therapeutic purposes. The purification of enzymes. The purification of enzymes involves a series of steps to isolate the enzyme of interest from other cellular components and contaminants. The purification process typically includes several techniques that exploit the unique properties of enzymes such as their size, charge, solubility, and specific enzymatic activity. Here are common steps involved in the purification of enzymes. Cell disruption. The process begins by breaking open the cells containing the enzymes. This can be achieved through mechanical methods such as example homogenization or grinding, enzymatic digestion, or by using specialized equipment such as cell disruptor. Centrifugation. Cell debris and organelles and other larger particles are removed by centrifugation. The cell lysate is spun at high speeds in a centrifuge and different components depend on their size and density. Fractionation. Additional separation steps are employed to fractionate the cell extract based on properties such as solubility, charge, or molecular weight. Techniques may include precipitation, ultrafiltration, or differential centrifugation. Purification of enzymes continued. So chromatography techniques can be used. In chromatography, there are various types of chromatography: ion exchange chromatography, size exclusion chromatography, affinity chromatography. So ion exchange separates proteins based on their charge. Size exclusion chromatography, there is gel filtration, separates proteins based on size. Affinity chromatography takes advantage of the specific binding between the enzyme and the ligand attached to the chromatography matrix. The enzyme is selectively retained and then eluted. Precipitation, ammonium sulfate or other precipitating agents can be used to selectively precipitate proteins. Enzymes with higher solubility under specific conditions remain in solution while contaminants are removed. Ultrafiltration, dialysis, used to exchange buffer conditions, remove small molecules or concentrate the enzyme. This is particularly useful when changing the buffer system before the next purification step. Assay and monitoring. Throughout the purification process, the enzyme activity is monitored using specific assays. This ensures that the enzyme is still active and helps to track its concentration and purity. Analysis. The final purified enzyme is analyzed for purity and molecular weight using techniques such as SDS page, sodium dodecyl sulfate, polyacrylamide gel, electrophoresis, or mass spectrometry. Enzyme immobilization. Enzyme immobilization refers to the attachment or confinement of enzymes to a solid support or within a matrix, restricting their movement in a particular space. Immobilization offers several advantages in various applications, including enhanced stability, reusability, and easier separation of enzymes from reaction products. Here are some common methods and applications of enzyme immobilization. Like physical adsorption, covalent bonding, entrapment, encapsulation, cross-linking, immobilization on membranes. Physical adsorption means enzymes are adsorbed onto a solid support through non-covalent interactions such as hydrogen bonding, van der Waals forces, and hydrophobic interactions. This method is simple but may result in enzyme leaching. Covalent binding. Enzymes are covalently attached to a solid support using chemical reactions, forming strong and stable bonds. This method provides better stability but may affect enzyme activity. Entrapment. Enzymes are physically trapped within a gel or matrix, restricting their movement. This method retains enzyme activity but may suffer from mass transfer limitations. And encapsulation. Enzymes are enclosed in a permeable membrane or microcapsule, protecting them from external factors. Encapsulation can enhance stability and protect enzymes from harsh conditions. Cross-linking. Enzymes are chemically cross-linked to form stable three-dimensional structure. Cross-linking improves stability but may affect enzyme activity. Immobilization on membranes. Enzymes are immobilized on membrane surfaces, allowing for continuous flow processes. A membrane-based immobilization provides advantages in separation processes. Materials used in enzyme immobilization: polymers, polyacrylamide, polyvinyl alcohol, polyurethane, alginate hydrogels. These are organic. Inorganic supports: silica, alumina, zeolites. Next, cytosine, carbon nanotubes, nanoparticles, agarose and agarose derivatives. All these materials are used in enzyme immobilization. and ion exchange resins, magnetic nanoparticles, glass beads or fibers, cellulose and derivatives, magnetic particles. All these are used in enzyme immobilization applications. So biocatalysis, biomedical applications, wastewater treatment, bioremediation, analytical chemistry, food industry. The choice of immobilization method depends on the specific application, the property of the enzyme, and the desired outcome. Researchers continue to explore new immobilization techniques and materials to optimize the performance of immobilized enzymes in various fields. Industrial production. Downstream processing of polysaccharide degrading enzymes by affinity chromatography. So this is the process by which the enzyme is purified. Industrial production of enzymes. So the reactor is looking like this. So is a specialization of bioprocess engineering. Is a specialization of chemical engineering. It deals with design and development of equipment and processes for manufacturing of products such as agriculture, food, feed, pharmaceutical, nutraceuticals, chemicals and polymers, and paper from biological materials, and treatment of wastewater. How enzymes work and what are the industries? Enzymes work by lowering the activation energy of the reaction, and these are the industries in which the enzymes are worked. Enzymes in industries. And example of an enzyme is papain. Papain is an enzyme extracted from the latex of the papaya fruit. It is a protease, meaning that it is capable of breaking down proteins into smaller peptides or amino acids by hydrolyzing peptide bonds. Papain is widely used in various industries due to its proteolytic properties and has several applications. So, and it also has its applications in these industries. And this is the end of the slide. Thank you.